पाहिजे तसे रिझल्ट नाहीत भेटत. तिचं असं म्हणणं राहते की ECLला आपण भरपूर करावं लागतं आणि भरपूर केल्यानंतर पण चांगल्या लेवलमध्ये नाही होत. का चूज केलं तुम्ही सगळं? त्यांनी का चूज केलं? काम खूप जास्त आहे, मेहनत खूप जास्त आहे, पण रिझल्ट आल्यानंतर मोठे एक्सपेरिमेंटचे नावं ऐकून मेहनत जास्त बाकीचे करतात ते नाही आहे. तसं नाही म्हणून वेगवेगळं करतात. कोणीचे पण प्रो आणि कोण असं नाही की एक लय भारी नाही, एक म्हणजे एकाच खूप स्ट्रगल आहे नाही, दोन्हीमध्ये पण स्ट्रगल आहे, पण त्याचे प्रो आणि कॉन्स प्रत्येकाचे. जसं की या लोकांना सगळं कल्चरवर डिपेंड राहावं लागतं ना, ते ग्रोथ झालं नाही झालं. त्यांचे कधी कधी एक्सपेरिमेंट केलेले कल्चर ग्रो होत नाही, तर सगळं व्हायला जातं एवढं केलेलं. मेन गोष्ट म्हणजे ब्लड भेटणं वगैरे त्या गोष्टीत ना, आपल्या स्ट्रेन जनरेट केलं ते एक महिना नाही केलं तर मग काय एवढं डिफरन्शिएट करून ठेवलं मला कळतच नाही. एवढं काही नाही आहे डिफरन्शिएट.
Good evening everybody. We'll wait 5 more minutes and then start the session.
Okay, ma'am.
hope I'll be audible to everyone.
Yes, ma'am.
Okay, so let's start the session for today. Okay, so in the last class also we were discussing about kidney and its function, okay, and how it does the filtration of the blood and how is the structure is made. Okay, so this is a diagrammatic image for so this is an image which shows that so this is the afferent arteriole, okay, which is taking the blood from the circulation to the kidney, and afferent arteriole gets open or situated here in a Bowman's capsule, where it forms the get divided into certain capillaries which are glomerulus, and then here the actual filtration of the blood takes place, okay, and the blood gets filtered and enters into the Bowman's capsule, this capillary space, okay, and the remaining blood will go in the with the afferent arteriole, okay, and whatever component of the blood or the plasma is filtered, it comes into this capillary space and then moves further in the later part of the Bowman's capsule or this nephron, okay, so where the proximal convoluted tubule start, okay, so if we'll so this is the entire one nephron. If I'll show you, so this is the afferent arteriole which is bringing the blood to the glomerulus, where there are glomerular capillaries, okay, where there are certain types of special cells are present, if you remember those podocytes, okay, which are present only in these kind of capillaries, okay, and here the blood gets filtered where it comes to the capillary space, and then and then from here in the Bowman's capsule, when the filtrate comes, then it goes to this particular tubule which is called as proximal tubule, okay, the proximal convoluted tubule further goes in the down and then forms a tube which is referred as loop of Henle, okay, so loop of Henle is a thin descending limb and then thin ascending limb, and then it has a thick ascending limb, okay, then it forms a distal tubule, distal convoluted tubule, which has a opening in the this collecting duct, okay, so usually, so in the kidney structure also, if you remember, we talked about renal cortex and renal medulla, okay, so we talked about renal cortex and the renal medulla. So this upper portion, if you will see, so majority of the portion comes in the renal cortex part, and then this loop of Henle majorly comes in the renal medulla part, okay, so when the blood gets filtered, okay, and after the filtration, whatever amount of the filtrate is passes through this loop of Henle and distal convoluted tubule, simultaneously there is a reabsorption and excretion of substances, okay, and then whatever goes to this collecting duct, that makes the urine, okay, before that it's a filtrate where reabsorption and excretion of ions, water, salts will take place, okay, where they go to the circulation again, okay, let's first see this, okay, so if you'll see this image, so here it is, the filtration is taking place, okay, which is glomerulus and Bowman's capsule, okay, then the reabsorption starts, reabsorption of these particular ions, amino acid, glucose, okay, so if you remember, we talked that all the amino acids and glucose gets reabsorbed, okay, and they do not come into this filtrate or in urine. Okay, so here in the proximal convoluted tubule you will see the sodium ion, potassium ion, water, glucose, amino acid, majority of them are getting reabsorbed. Okay, then there is a loop of Henle where the concentration of the salts are taking place. Okay, here in this particular area, and then secretion of certain ions like potassium ion, hydrogen ions, secretion of into the filtrate, and then again in the collecting duct reabsorption of water takes place. Okay, to make the urine more concentrated, and then again it goes to the final thing goes to the collecting duct and forms the urine. Okay, so reabsorption and secretion by the renal tubules. As the glomerular filtrate enters the renal tubules, it flows sequentially through the successive parts of the tubule. So the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Okay, so before it excreted as urine, and these are the epithelial cells if you will see. Okay, so the after the Bowman's capsule we have the proximal convoluted tubule. You see these cells are very much thick. Okay, and the lumen is very small in diameter. Okay, then comes the loop of Henle. Okay, so these thin portion of loop of Henle, okay, where the cells are thin and giving enough space of the lumen. Then we have the distal convoluted tubule, comparatively thicker cells than the loop of Henle, and then we have a collecting duct which is having the highest diameter. Okay, next. Okay, so this is the part. So these are the cells. Suppose in the kidney these are the tubular cells. Okay, so these are the tubular cells. Okay, here is the lumen which we just now see. Okay, so the lumen. Okay, so if you like take one cell, okay, only this portion, and one side it is facing lumen, another side is it is facing the interstitial fluid. Okay, in that way if you will see, so this cell, this side it is facing the lumen and this side it is facing the interstitial tube and the blood capillaries. Okay, just a minute. So then filtrate is coming to the lumen. Okay, and then excretion and going for the excretion. Here in this particular direction the reabsorption takes place. That means from the filtrate the substance are getting reabsorbed. Okay, so there are certain types of transport. Okay, so one is the active transport where the ATP is involved, and then we have the passive transfer which is by the diffusion. Okay, and also the transport of the water by osmosis. Okay, because the osmotic gradient is created. Okay, so these are the main things which we will be studying today. Okay, and when it comes to the interstitial fluid it goes to the blood and reabsorbed. So whatever filtrate is coming, okay, there are majority essential things body tries to take back from that filtrate. Okay, for example the sodium ions. Okay, the sodium is very much important substance, and that is why its reabsorption is very much important. So there are several mechanisms the nature has made in our body that tries to reabsorb as much as sodium that as it can. Okay, so for a substance to be reabsorbed it must be transported. Okay, one across the tubular epithelial membranes into the renal interstitial fluid. Okay, from here to this renal interstitial tubule, and then through the peritubular capillary membrane back to the blood. Okay, for instance water and solutes can be transported either through the cell membrane themselves or through the junctional spaces between the cells. Then after absorption across the tubular epithelial cell into the interstitial fluid, water and solutes are transported to the rest of the way through the peritubular capillaries walls into the blood, that is mediated by hydrostatic and colloid osmotic forces. Okay. So reabsorption is secretion along different parts of the nephron. Okay. So after the blood is filtered from the Bowman's capsule, it first enters to the proximal convoluted tubule. Okay, the proximal tubule, I can say. So let's start with that. So this is a section of proximal tubule. These are the cells. Okay, so normally about 65% of the filtered load of sodium and water and slightly lower percentage of the filtered chloride are reabsorbed by the proximal convoluted tubule before the filtrate reaches to the loop of Henle. Okay, so these percentage can be increased or decreased in different physiological condition. Okay, so here you can see a lot of sodium ions, chloride ion, potassium ion, water, glucose, amino acid, these are reabsorbed from these cells. Okay. Okay. Skip this part. Yeah, so here also you'll see this part is the lumen part and this is the interstitial fluid part. Here if you will see there is a pump. So the part which is the cells that portion which is facing towards the lumen, that is referred as apical or that can be referred as apical membrane or apical portion. Okay, and here this portion is referred as basal membrane. Okay, so this is the apical membrane, this is the basal membrane. So in the basal membrane or the basal side we have this sodium potassium ATPase pump. Okay, so this is an active transport because for the transport of sodium to the interstitial fluid the ATP is required, so it is an active transport. Okay, as I've already mentioned, sodium is a very important substance that is needed for body for different several kind of functions. So that is why via this active transport body in the basal lamina, the body tries to reabsorb the sodium. Okay, so as the sodium reabsorption happens, it creates a gradient here for of low sodium. Then the filtrate which is carrying the sodium gets reabsorbed and comes to this cells or tubular cells. Okay, so here you can see, so this is the we are seeing the proximal convoluted tubule. So this line showing the primary active transport. Okay, and because of that the secondary active transport is happening. So as I have said, sodium is driven back to the interstitial fluid by the active transport because ATP is involved, and when the sodium concentration decreases in the cell, it takes from this lumen filtrate in the lumen. Okay, so then this via the passive transport, because of the diffusion, the sodium comes, and when the sodium comes it also brings glucose along with it via the symport. So because of this active transport, the another molecule which is coming, and that is called secondary active transport. Okay, so with the sodium glucose is also coming. Okay, and everything else is a passive transport or by the diffusion. Urea, water, glucose, amino acid, they all are coming by the passive transport or the ion channels. Okay, so sodium reabsorbed by primary active transport enters epithelial cell passively, pumped to blood, balancing potassium leaves net negative charge. Okay. Let's see. So the pumping of sodium out of the cell across the basolateral membrane of the cell favors passive diffusion of sodium across the luminal membrane of the cell from the tubular lumen into the cell for two reasons. So there is a concentration gradient favoring sodium diffusion into the cell because intracellular sodium concentration is low and the tubular fluid sodium concentration is high. Okay, because in our blood usually the sodium concentration is 140 milliequivalent per liter. Okay, so when in the glomerulus the blood is filtered. Okay, so majority of the plasma comes in this filtrate. and in our plasma the sodium concentration is 140 milliequivalent per liter. So here the sodium concentration is 140 milliequivalent per liter. However, because of the active transport in this cell, the sodium concentration becomes 12 milliequivalent per liter, and that is why the passive transport or via the diffusion, the sodium enters from the fluid to the lumen of this tubular cells. Okay, and again actively transported back to the interstitial fluid. And also there is one more reason, that negative minus 70 millivolt intracellular potential attracts the positive sodium ions from the tubular lumen into the cell. Okay, so again this is the blood side, this is the lumen side. Lumen side we are referring as the apical side, and the blood side or the interstitial fluid side is the basal side. Okay, always remember the sodium potassium ATPase pump is only present on the basal side. Okay, it is never present in the apical side, it is always present in the basal side. Okay, so this is the same thing again, it is the tubular lumen, interstitial fluid, it is showing the transport, active transport, and along with that the passive transport is being shown, and this is the counter transport again. So these are the pumps which are primarily active transporter that are known: sodium potassium ATPase pump, hydrogen ATPase pump, hydrogen potassium ATPase pump, and calcium ATPase pump. Has been talked already. Start from here. Okay, so secretion of organic acids and bases by the proximal tubule. So here so these are the some salts, oxalates, urates or drug, okay, that are also being secreted in the proximal convoluted tubule so that they can be avoided. Okay, so renal excretion of drugs, filtration of small non-protein bound drug occurs through glomerular capillary pores. Lipid soluble and unionized drugs are passively reabsorbed, and active secretion of organic acids and bases occurs only in the proximal tubular segment. So here you can see from the efferent arteriole which is coming here, okay, the peritubular network, it is again secreting the drugs into the filtrate. Okay, that is making a rapid clearance of the drugs that have been taken. Okay, so there is
Peritubular works. Once again, you will repeat.
it is from afferent arteriole the drug is coming to the body. Okay, and if it is not filtered here in the glomerulus, the drug is again going back to the efferent arteriole. Okay, but our body tries to remove the drug as much as possible. So when again from the efferent arteriole, when it is making a peritubular arteries here, the capillaries here, the drug is via the active secretion, okay, by binding to the certain protein molecules or receptors which is present in the proximal convoluted tubule, it enters to the filtrate, and when it enters to the filtrate or the secretion of the drug is happening from this peritubular artery to the proximal convoluted tubule, and then it goes to the excretion part or in the urine, and it gets eliminated from the body. Okay, so that is causing a rapid clearance of the drug. Okay, so in the case of certain drugs such as penicillin and salicylate, the rapid clearance by the kidney creates a problem in maintaining a therapeutically effective drug concentration. For example, for some drugs the kidney clears them out very quickly, so their effect is not made on the body properly. So that is why this is a common problem, the drug clearance by the kidney. That is why the drug the more effective drug is still in a demand for several different kind of disease. Okay, so then we have a concept of aquaporins. Okay, so as we are mentioning that water is also being reabsorbed. Okay, so when the filtration is happening, lot of water is also coming to the filtrate. So our body try to retain as much water as it can. So for the retention of water, there are channels present in the membrane of the renal tubules. Okay, and those channels are nothing but aquaporins. So aquaporins selectively conduct water molecule in and out of the cell while preventing the passage of ions and other solutes. So water molecule traverses through the pore of the channel in single file. The presence of water channels increases membrane permeability to water. Okay, so if this is the one nephron you are seeing Bowman's capsule, proximal convoluted tubule, loop of Henle, then again distal convoluted tubule, and this is the collecting duct. Okay, so if you will see these colorful dots are the different aquaporins. Okay, so these are different aquaporins. You will see many various of aquaporins are present in the renal tubules. So but they are not present everywhere on the renal tubule. So where they are present, if you see proximal convoluted tubule, entirely they are present. Then the descending loop of Henle, they are present. Then ascending loop of Henle and thin ascending loop of Henle and thick ascending loop of Henle, and in the distal convoluted tubule these aquaporins are not present. Okay, so this entire portion is devoid of the aquaporins, so water reabsorption from this portion does not take place. Okay, and then again in the collecting duct we have the aquaporins. Okay, so water movement will happen only in the proximal convoluted tubule and the descending loop of Henle. Okay, here it is written, the descending limb of loop of Henle is permeable to water due to presence of aquaporin 1 in both apical and basolateral membrane, but the ascending limb is impermeable to water. Okay, both thin and thick ascending limb, and also the distal convoluted tubule. So let's see. Okay, solute and water transport in the loop of Henle. So in the proximal convoluted tubule we have seen majority of the ions and the glucose and amino acids are being reabsorbed, and also water. Okay, then the filtrate is coming to the loop of Henle. Okay, so here this green highlighted portion we are talking about this. So this descending loop of Henle is having aquaporins, and this is a thin cells or the lumen of thin cells. Okay, so the loop of Henle consists of three functionally distinct segments. Okay, so thin descending segment, thin ascending segment, or one thin thick ascending segment. Okay, so the descending part of the thin segment is highly permeable to water and moderately permeable to most solutes. Okay, about 20% of the filtered water is reabsorbed in the loop of Henle, and almost all of this occurs in the thin descending limb because that is only permeable to the water. Okay, and in the ascending limb, including both thin and thick portions, is virtually impermeable to water because aquaporins were not present there. So a characteristic that is important for concentrating the urine. Okay, so here when the water is reabsorbed, okay, so water is reabsorbed here, then in this portion what happens, a lot of salts are being concentrated. Okay, so here we'll see the thick segment of loop of Henle, which begins about halfway up the ascending limb, has thick epithelial cells. These are the thick epithelial cells that have high metabolic activity and are capable of active reabsorption of sodium chloride and potassium. So about 20% of the filtered loads of sodium chloride and potassium are reabsorbed in the loop of Henle, mostly in the thick ascending limb. Considerable amount of other ions are also reabsorbed. So from this portion 25% of the ions are being reabsorbed. Okay, the thick segment of the ascending loop of Henle is virtually impermeable to water. So the tubular fluid in the ascending limb becomes very dilute as it flows towards the distal tubule, because here what happens, the reabsorption of the salts is happening. Okay, and water is not being reabsorbed. So overall if you'll see, whatever fluid was there, sodium concentrations has decreased, so that's why this filtrate becomes very much diluted. Okay, so the tubular fluid in ascending limb becomes very dilute as it flows towards the distal tubule, a feature that is important in allowing the kidney to dilute or concentrate the urine under different conditions. Okay, so this we are seeing again the thick ascending tubule. So here we have in the basal side on this towards the interstitial fluid the active pump, sodium potassium ATPase pump, which is drawing sodium outwards in the interstitial fluid. Then we have this secondary active transport, again sodium is coming. Here the sodium is, this is not secondary active transport, sorry. So here again, because the osmotic gradient is created of less sodium in this cell, from this filtrate sodium is coming in this particular cell, and again sodium is being thrown out of this cell. Okay, then here we have one particular pump which is called NKCC. Okay, so sodium, potassium and CC, 2 chloride. Okay, sodium potassium chloride pump. Okay, so an important component of solute reabsorption in the thick ascending limb is sodium potassium ATPase pump in the epithelial cells of basolateral membrane, this one. Then this maintains a low intracellular sodium concentration. So the low intracellular sodium concentration in turn provides a favorable gradient for movement of sodium from the tubular fluid into the cell. Okay, so and in the thick ascending loop, movement of sodium across the luminal membrane is mediated primarily by one sodium 2 chloride 1 potassium co-transporter. So this co-transport protein carrier in the luminal membrane uses the potential energy released by the downhill diffusion of sodium into the cell to drive the reabsorption of potassium into the cell against a concentration gradient. Okay, so what is happening here? A lot of sodium is being reabsorbed because of the presence of these pump as well. Okay, here we have saw in the proximal tubule majority is being reabsorbed. Whatever is remaining in the loop of Henle, from there the water is being removed. Okay, so the water is being removed and lot of sodium ions and chloride ion and all those ions still remains, that make this fluid of very high osmolarity. Okay, of less water because aquaporins are there. Then as the urine is coming to this direction in the distal convoluted tubule, there is no aquaporins, so water movement is not happening, but there are these certain kind of channels. So in the thick ascending tubule, sorry, this is still loop of Henle we are talking about. So in the thick ascending loop of Henle, the presence of NKCC pump, okay, because of these transport, a lot of ions, sodium chloride, potassium ions are being reabsorbed and comes to the interstitial fluid. Okay, so so there are certain drugs, if I'll say, which are referred as loop diuretics. Okay, so in the loop of Henle, if you provide these drugs, so what these drugs do, it what does these drugs do? These drugs inhibit with this pump. Okay, how they bind to that portion where these chloride ions needs to be bind. Okay, so if the chloride ions are not bind, these pumps are not active, so these ions are not being reabsorbed. Okay, so that is why there is a problem or in the, what you say, in the reabsorption of these salts when these loop diuretics are given. So here you can see the NKCl co-transporter is a protein, okay, that aids in active transport of sodium potassium chloride into and out of the cells. So there are two varieties, NKCC1 and NKCC2. However, these are encoded by two different genes. Okay, so NKCC2 is specifically found in cells of thick ascending loop of limb of Henle in nephrons. Okay, So within these cells, NKCC2 resides in the apical membrane, here in this side, the lumen side. Okay, so urine in the thick ascending limb of the loop of Henle has relatively high concentration of sodium. Okay, so this is the electrochemical gradient of sodium favors movement of sodium from the urine into the cell. At this region, the nephron NKCC2 is major transport protein by which sodium is reabsorbed from the urine into the cell. So each molecule of sodium reabsorbed brings one molecule of potassium and two molecules of chloride. Sodium goes on to be reabsorbed into the blood. Okay, so there is a drug furosemide and other loop diuretics inhibit the activity of NKCC2, thereby impairing the sodium reabsorption in the thick ascending limbs of the loop of Henle, and impaired sodium reabsorption prevent the thick ascending loop from contributing to maintenance of blood pressure. Okay, so loop diuretics therefore ultimately result in decreased blood pressure. Okay, because because there is an inhibition in the reabsorption. Okay, and that is why the blood pressure is also ultimately decreased. This is the pump. Okay, so now we are talking okay. So then we have the early distal tubule. Okay, now after the loop of Henle, the filtrate will go to the early distal tubule. Okay, in the distal tubule, where in the early distal tubule. So early distal tubule also we saw there are no aquaporins. Okay, for the water reabsorption. Okay, and here this distal tubule again goes and talks to the glomerulus of the same nephron, where the juxtaglomerular cells are present that provides feedback control for the glomerular filtration rate that we have already discussed. Okay, so early distal tubule is highly convoluted and has many of the same reabsorptive characteristic of the thick segment of the ascending limb of loop of Henle. That is, it avidly reabsorb most of the ions including sodium, potassium and chloride, but is virtually impermeable to water and urea. For this reason, it is referred to as diluting segment because it also dilutes the tubular fluid. Okay, again from here the ions are being removed, and that is why the fluid is being diluted. Okay, so the water retains and the ions are removed, and that is why overall the filtrate is become diluted. Okay, so here we have, okay, so in the loop of Henle, in the ascending loop of Henle, thick ascending loop of Henle, we had those NKCC protein channels for the transport of sodium chloride and potassium. Here in this early distal tubule, we have this these particular pump or sodium chloride transporters, NCC. Okay, that was NKCC, this is NCC. Okay, so thiazide type diuretics act on this distal convoluted tubule and inhibit this particular symporter pump, NCC, leading to a retention of water in the urine, as water normally follows the penetrating solutes. Okay, so frequent urination is due to increased loss of water that has not been retained from the body. Frequent urination is due to the increased loss of water that is not being retained from the body as a result of a concomitant relationship with sodium loss from the convoluted tubule. Okay, mechanism. So we have here, these are thiazide of diuretics. These diuretics are type of thiazide, and the diuretics which are acting on this particular loop of Henle, those diuretics are called as loop diuretics. Okay, so both of these diuretics, what are their function is, they inhibit uh, uh, what to say, um, these particular pumps, okay, for the reabsorption of sodium ions. Basically, if I'll talk sodium ion, okay, so these diuretics, for example, in loop of Henle we have loop diuretics, and in distal convoluted tubule we have this thiazide diuretics. So these are diuretics because what they are doing here is they are inhibiting the reabsorption of the sodium ion. Okay, because these are the portion where the salts needs to be reabsorbed to the blood. Okay, but the reabsorption is being inhibited because of these particular inhibitors or drugs. Okay, and if the sodium is not going, if you remember in the very first slide only I have told, if the sodium is being reabsorbed, along with that, because of the osmotic gradient, water also follows this particular uh, ions. Okay, and that is why when the sodium is removed, water is also being removed. Okay, so that is maintaining the osmolarity. However, when the sodium is inhibited to be reabsorbed, then what happens, the water also being retained. Okay, so that makes the urine further diluted, or further frequent urination. Okay, so frequent urination is due to the increased loss of water that has not been retained from the body as a result of a concomitant relationship with sodium loss from the convoluted tubule. So these are the diuretics that has been used to dilute the urine. Then we have the, yeah, so this the early distal tubule which is inhibiting, so these are thiazide diuretics which is inhibit the reabsorption of this NCC pump. Then we have the late distal collecting tubule. Okay, so here also you can see in the late distal convoluted tubule we have basically two types of cell. One is the principal cell, another one is the alpha intercalated cells. Okay, so here we have ENaC. Okay, these are the transporters which are present in the lumen for, again, for the reabsorption of the sodium. Okay, so these are intercalated cells, the red one are the principal cells. So the principal cell in the connecting segment and the cortical collecting ducts are most important targets for aldosterone. Okay, so remember when we talked about in these particular section where we have the GFR controlled by the aldosterone hormone. Okay, so here what happens, there are a number of cells which were referred as juxtaglomerular cells. Okay, so those juxtaglomerular cells, in when they are sensing the low blood pressure, they releases the particular hormone which is renin. Okay, renin further produces the active component of the hormone angiotensin 2, which again increase the production of aldosterone. Altogether they maintain the blood pressure. Okay, so aldosterone target are the principal cells of the distal convoluted tubule. Okay, so aldosterone increases the sodium permeability of the luminal membrane and helps to reabsorb the sodium. So what does aldosterone exactly do? Okay, so this aldosterone, if you are seeing, so aldosterone, it from the um it comes to these particular cells. Okay, enters to these cells, and then what does it do? So aldosterone basically comes from the blood. Okay, so from the blood only it comes, and then when it enters the cell, it, what is they do, it goes to the nucleus and increase the production of these particular of the transcription factors which produces these particular ENaC what channels. Okay, for the sodium reabsorption. So sodium reabsorption is happening, sodium is being reabsorbed, along with that water is also being reabsorbed. Okay, water is also coming, and when the water is being retained, then the blood which is flowing with the low pressure, with the increased osmolarity, now it is flowing with the higher blood pressure. Okay, so that is how the aldosterone performs its function. Okay, so sodium reabsorption and potassium secretion by the principal cell depend on the activity of sodium potassium ATPase pump in each cell's basolateral membrane. This pump maintains a low sodium concentration inside the cell and therefore favors sodium diffusion into the cell. Okay, so this is the same thing that we are talking, epithelial sodium channel, ENACs, this is the name of the channel. Okay, so here you can see, so aldosterone which is coming to these particular cells, okay, from the blood, okay, from the blood it is coming and then entering, so it is a, and these are the receptors which are mineralocorticoid receptors. Mineralo because it is ultimately it is working on the minerals with sodium, calcium, potassium. Okay, and cortico is the receptor. Okay, so mineralocorticoid receptor because it is coming from the coming to the cortex. Okay, so this is the receptor where this aldosterone is binding, and aldosterone again going and activating the transcription factors which are helping the production of ENAC and also the sodium potassium ATPase pump. Okay, which is making the sodium reabsorption more and more in the blood from the lumen or from the filtrate. Okay, so altogether that is causing the blood pressure to increase. Okay, so these are the intercalated cells, avidly secreted hydrogen reabsorbed bicarbonate and potassium ions. Okay, so in the principal cell that we have the intercalated cell, they are mediating the bicarbonate and potassium ions transport. Okay, so this is the late distal tubule. The permeability of the late distal tubule and the cortical collecting duct to water is controlled by the concentration of ADH, okay, antidiuretic hormone. Okay, so with high levels of ADH, these tubular segments are permeable to water, but in the absence of ADH, they are virtually impermeable to water. Okay, so this special characteristic provide an important mechanism for controlling the degree of the dilution or the concentration of the urine. Then again from here the reabsorption is happening. So the final site for processing the urine and therefore play an extremely important role in determining the final urine output of the water and solute. Okay, so the permeability of the medullary collecting duct to water is controlled by the level of ADH. So unlike the cortical collecting tubule, the medullary collecting duct is permeable to urea. So therefore some of the tubular urea is reabsorbed into the medullary interstitium. So this is the entire graph. If you will see, the proximal tubule, loop of Henle, distal convoluted tubule and the collecting tubule. So everything, if we are starting from 1, then in the filtrate, okay, so this is the tubular fluid plasma concentration. So the concentration of sodium remains and potassium remains the same, then it decreases, then again it come to the normal level. Okay, however, glucose, protein, amino acid, all these concentrations are being decreases in the proximal tubule only, because in the proximal only all the glucose, amino acid, proteins are being removed from the filtrate. Okay, so the concentration of these becomes very low. Okay, and then in the sodium and potassium ion concentration also becomes low in the filtrate because of the reabsorption, continuous reabsorption. However, the concentration of creatinine and other urea, these increase because the secretion is happening and there is no reabsorption for certain drugs. So that is why they gets their concentration in the filtrate is increasing and that gets excreted with the urine. Okay. So this is the angiotensin 2 also increases the sodium and water reabsorption, because angiotensin 2 stimulates the aldosterone secretion, which in turn increases the sodium reabsorption. Okay. this we have already talked. Angiotensin 2 stimulates sodium reabsorption in the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule and the collecting tubule. One of the direct effect of angiotensin 2 is to stimulate the sodium potassium ATPase pump on the tubular epithelial cell basolateral membrane, okay, the ATPase pump. And the second effect is to stimulate sodium hydrogen exchange in the luminal membrane, especially in the proximal tubule. A third effect of this hormone is to stimulate the sodium bicarbonate co-transporter in the basolateral membrane. So this is very important for the sodium reabsorption. Okay. This is again overall view if we'll see. So we have this atrial, so this we will discuss maybe. We have angiotensin 2, which is synthesized in response to low blood pressure and causes efferent arteriole constriction, which is increasing the GFR, okay, and compensatory sodium reabsorption in the proximal and distal nephron, and net effect preservation of the renal function, okay. Then we have this particular section of proximal convoluted tubule where sugar, amino acid and sodium is being reabsorbed, majority of them. Then we have the loop of Henle. Here in the ascending loop of Henle we have the NKCC pump, okay, which is permeable, making the salts to come out of this lumen, okay, and this pump is affected by the loop diuretics, okay, which is inhibiting the reabsorption of these substrate, okay. And then we have here the calcium magnesium reabsorption. Again here we have the sodium chloride, the NCC pump in the distal convoluted tubule, and again here also the aldosterone is coming to the action, secreted in response to low blood volume and increased plasma potassium ion, and causes sodium reabsorption and potassium secretion and hydrogen secretion. Then this is the ADH or the vasopressin hormone, secreted in response of higher plasma osmolarity and low blood volume, and binds to the receptor on the principal cells, causing increased number of aquaporins and increased water reabsorption, okay. So aldosterone, basically what does it do in the distal convoluted tubule? It is increasing the ENaC or the carriers or responsible for the sodium reabsorption. However, ADH, what does it do in the higher plasma osmolarity? It implements certain aquaporins in the collecting duct, which causes the water reabsorption. So regulation of extracellular fluid osmolarity and sodium concentration. So there are certain mechanisms. So the mechanism that causes the kidney to eliminate the excess water by excreting or dilute urine, the mechanism that causes the kidney to conserve water by excreting a concentrated urine, okay. So the renal feedback mechanism that control the extracellular fluid, sodium concentration and the osmolarity. The thirst and the salt appetite mechanism that determine the intake of water and salt, which also help to control extracellular fluid volume, osmolarity and sodium concentration. So let's see that. So here, so we have seen that in the proximal convoluted tubule and the descending loop of Henle, and in the later portion of the collecting duct, only those contains the aquaporins, okay. So here, so the osmolarity of the blood, if we consider it's 300 milliosmol per liter, okay. So when the blood enters and gets filtered, it comes with a 300 milliosmol osmolarity. So from here the water reabsorption is taking place, okay. So from 300, if you'll see, it has gone to the 400, okay. Then again water reabsorption is happening until here it becomes very much concentrated with the salts and the ions. So the osmolarity of the water is increased because of the high sodium water reabsorption, okay. Then as we go to the ascending limb of the loop of Henle, then the sodium and the salts reabsorption takes place. Okay, so then again salts are getting reabsorbed. Okay, okay, so the salts are being reabsorbed, and then again it reaches to so with the water, so water is not being reabsorbed, the salts are being reabsorbed, then again the osmolarity decreases and it has come to the 100. Okay, so then again in this portion, in the distal convoluted tubule, again salt reabsorption is happening, then 100 osmolarity is maintained. Then again here also, so if we are seeing two different situation, okay, so the kidney excrete excess water by forming a dilute urine. Okay, so if you have taken or you have have more intake of water, okay, if you have done more intake of water, excess of water is there in your body, so your body's response will be make a dilute urine. Okay, make a dilute urine, and so the formation of concentrated urine when ADH is high. Okay, so the normal kidney has tremendous capability of vary the relative proportion of solutes and water in the urine in response to various challenges. So when there is excess of water in the body fluid osmolarity is reduced. Okay, the kidney can excrete urine with an osmolarity as low as 50 milliosmol per liter. Okay, a concentration that is only about 1/6 of the osmolarity of the normal extracellular fluid. So conversely, when there is a deficit of water, okay, and the extracellular fluid osmolarity is high, the kidney can excrete urine with a concentration of 1200 to 1400 milliosmol per liter. Okay, so when there is an um, um, what to say, deficiency of water, okay, then in that deficiency of water this particular hormone is being released, which is antidiuretic hormone. Okay, so antidiuretic hormone makes the urine concentrated because it allows water reabsorption, because you are having low on water. So if the water is low, your body will sense that signal, with that signal antidiuretic hormone is being released. So here in this portion, so till here everything is fine. Okay, here if you will see in the collecting duct, so we have discussed the antidiuretic hormone uh, uh, puts the aquaporins in the collecting duct, which causes the reabsorption of water, because your body is also already at a low water concentration, so it tries to take as much water from the filtrate or from the urine and try to make a very concentrated urine, so your body loses very less water. So here the urine concentration or the osmolarity has come down to the 1200 milliosmol per liter. However, when there is a low ADH in the urine, so the osmolarity generally is 70-50 milliosmol per liter. Okay, so equally important, the kidney can excrete a large volume of dilute urine, okay, or a small volume of concentrated urine without major changes in the rates of excretion of solutes such as sodium and potassium. This ability to regulate water excretion independently of the solute excretion is necessary for survival, especially when fluid intake is limited. Okay, so this is a renal mechanism for excreting a dilute urine. Okay, so suppose you have taken excess of water in this particular concentration, so usually your blood osmolarity is 300 milliosmol per liter. Okay, so when you have excess of water in the filtrate, you can see the high osmolarity is there. Okay, and then it goes down. Okay, it goes down and the urine osmolarity has again come high, and the plasma osmolarity is almost remain at the constant. Okay, and This is the urinary solute excretion and urine flow rate. So the urine flow rate is increasing, okay, the urine flow rate is increasing with the time, and again bringing it back to the normal urine osmolarity, and no changes in the plasma osmolarity. The kidneys conserve water by excreting a concentrated urine that we have seen. So our body can concentrate the urine up to 1200 milliosmol per liter, but this particular desert animal, this Australian mouse, it can concentrate it using urine up to 10,000 milliosmol per liter, okay, for the survivor. And however, the other one, this one, it can concentrate the urine only about 500 milliosmol per liter. Okay, then we have this osmotic gradient in the renal medulla. So if you will see, this is the cortex, this is the medulla. Here our body's osmolarity, blood osmolarity, which is 300, and as we are coming towards close to the medulla in the center, the osmolarity is gradually increasing, okay, 400, 600, 900, 1200, okay. So this osmolarity you are able to see in the interstitial fluid. Okay. So the counter current mechanism produces a hyperosmotic renal medullary interstitium, okay. So if we'll see this one, so this is called generation of single effect in the loop of Henle. So this is the initial condition, which is the interstitial fluid. This is, so the loop of Henle we are taking from here, this, the descending limb and the ascending limb, okay. So this is the interstitial fluid. The walls of the ascending limb are essentially impermeable of the water and are drawn thick to symbolize that, okay. So this portion is impermeable to water, that is why the lining is thick, okay. So then this is the process that generate that single effect. So active transport of sodium chloride out of the fluid into the ascending limb, this one, ascending limb dilutes that fluid and concentrate the interstitial fluid, okay. The walls of the descending limb are permeable to water, so the fluid inside becomes concentrated by losing water, the water osmolarity to the interstitial fluid, and sometimes by gaining sodium and chloride by the diffusion, okay. So this is how this is being maintained. So the single effect is the difference in the osmotic pressure and the sodium chloride concentration between the ascending limb fluid and the adjacent interstitial fluid and the descending limb fluid, okay, because of the transport. So this is the loop of Henle, that U shape, okay, that loop of Henle. So here initially the filtrate is coming with the 300 milliosmol osmolarity, okay. Then as we are seeing here, this those transporters are present, okay, that reabsorbs the sodium, okay, that is making this particular portion of the filtrate of less osmolarity. The osmolarity is being decreased, okay, 200, okay. So from 300 it is becoming to 200, okay, and outside it is becoming 400, that's interstitial fluid, okay. So again from with the interstitial fluid, this particular filtrate or the liquid tries to comes to the balance, okay, by water reabsorption, water removal, okay. Then again both have come to a similar osmotic concentration, interstitial fluid and the descending limb 400 osmolarity, because water reabsorption is happening, okay. But the fluid is moving, then again this 400 milliosmol liquid comes to the ascending loop, then ascending loop also it is going, okay. Then again this 400 is coming here, then again from this one the reabsorption of salt will take place, decreasing the osmolarity in this region again, okay. Again this is the interstitial fluid osmolarity increases, and again with the water reabsorption and the sodium and the osmolarity maintenance, This step repeats 5 to 6 times, and that is how a gradient is being created in the interstitial fluid, which we have seen in the cortex 300, then 700, 1000 and up to 1200. Okay, so this is a counter current multiplier system in the loop of Henle for producing a hyperosmotic renal medulla. Okay. Also the vasa recta, this particular artery, these are the also play a very important role. So the vasa recta preserve the gradient, they act as counter current and exchangers. Okay, so from here you will see the osmolarity 300, 300, 400, 600, 900 and 1200. Then so this is the one which is descending, and along with that if you will see this vasa recta is coming which is ascending. Okay, so this capillary is ascending, so here the liquid is flowing in this direction, and here the liquid is flowing in the opposite direction. So the long nephron loops of the juxtamedullary nephrons create the gradient, and they act as a counter current multipliers. Okay. This one I was talking. Yeah, so these and these, this one is on a close proximity, so this is bringing the fluid down, and this is bringing the fluid the blood, the filtered blood up. Okay, so this is a counter current mechanism which is also helping in maintaining this osmolarity gradient in the medulla. So this is the interstitial fluid osmolarity. Okay, and this is the vasa recta, which is also maintaining the same molarity with this. So the vasa recta, like other capillaries, are highly permeable to solutes in the blood except for the plasma protein. So as the blood descends into the medulla towards the papillae, it becomes progressively more concentrated, partly by solute entry from the interstitium and partly by loss of water into the interstitium. So by the time blood reaches the tips of the vasa recta, it has concentration of about 1200 milliosmol per liter, the same as of the medullary interstitium. As blood ascends back towards the cortex, it becomes okay. Okay, so let's see. So here also the total concentration of the organic osmolytes from cortex to the medulla, it is increasing. So the total concentration of the 4 organic osmolarity within the medullary cell increases with increasing depth. Okay, so this is the osmoreceptor ADH feedback mechanism. Okay, so is there a fall in the blood pressure, and how is blood alerted? Okay, so here we have water deficiency. Because of the less water, okay, the blood pressure falls. Okay, and then that osmolarity is also being sensed. Okay, so here is the mechanism given. So in the pituitary gland, from the below the hypothalamus, we have this neurons which is SON PVN. Okay, these are the neurons which releases the hormone which is vasopressin. Okay, so the vasopressin receptors activates. Okay, so because of the water deficiency, extracellular osmolarity increases. Okay, so when the extracellular osmolarity increases, because water is less and the salts are more. Okay, so the osmolarity increases, extracellular osmolarity, then the osmoreceptors gets activated. Because of the osmoreceptors activation, the ADH secretion is happening in the posterior pituitary. Okay, so the ADH, antidiuretic hormone, or the vasopressin, is being released from the posterior pituitary. So in the plasma, so this hormone is coming in the circulation. Okay, via the circulation. because in the pituitary gland here only there are capillaries present which are receiving the antidiuretic hormone. It comes in the circulation in the plasma ADH. So in the plasma the antidiuretic hormone concentration increases. Okay, so with the increase of the antidiuretic hormone, the water permeability in the distal tubules and the collecting duct increases. Okay, because we have talked that and it causes the insertion of aquaporins in those areas collecting ducts, so the water permeability increases, so water reabsorption increases, and water reabsorption increases, then we have the okay, and the water reabsorption increases, which counterback or with the reabsorption more water comes into the blood, and that makes the blood a little dilute and increases the pressure, okay, the blood pressure. And when the water is excreted, then it again make a negative feedback of the water deficit. Okay, so this is what shown here. So the osmoreceptor activation, it also activates thirst. With the thirst, drinking water is being is activated, that sense is given. Okay, and with the drinking water, the plasma osmolarity is giving to the negative signal. Then again the activation is happening, AVP secretion, plasma vasopressin receptor activation is happening, water reabsorption is happening, and it is giving negative signal to the plasma osmolarity. Okay, and this is a cell autonomous osmoreception in vasopressin neurons. Okay, so these are the neurons. Usually as a set point it is minus 60 millivolt. Okay, a basal level of vasopressin is being released. Okay, so when there is a change in the osmolarity, okay, so the osmolarity is being changed. Here you can say osmoreceptor activation, osmolarity is being changed. So what happens with the hypertonic, suppose low water, because of the water deficit hypertonic solution is being formed that makes more or making the sodium to concentrate more sodium. So with the more sodium it enters, and with the positive charge the depolarization of this neuron is happening. With the depolarization more signals are coming. Okay, more depolarization, more signals are coming, so increasing in the vasopressin amount. Okay, and it is working as an antidiuresis. Okay, that is making the blood concentrated and reabsorbing the water, so that this hypertonic sodium or high osmolarity or sorry yes, high osmolarity can come back to the set point. Okay, then at the opposite concentration the membrane is becoming what to say the minus 65 millivolts. Okay, so it is becoming hyperpolarized, and that is why the frequency is reduced, the frequency of action potential is reduced, and because of that low vasopressin is released. And when the low vasopressin is released, the water reabsorption is less. Okay, because of the less so low vasopressin, and that is why again the plasma osmolarity is come back to the set point. Yeah, so this is the dilute urine, this is the concentrated urine. So in the dilute urine the plasma vasopressin concentration, in that case it is high, and when there is a concentrated urine the vasopressin concentration is very much sorry, this is about this is the rate. Okay, this is the relationship among plasma vasopressin concentration and rate of the water excretion and the solute excretion. Okay, so this is the water excretion, this is the which is showing. So the water excretion rate has become more when the vasopressin is less. Okay, when the vasopressin is less, diuresis is happening because water needs to be removed. Okay, and here when vasopressin is more, water excretion is less, and concentrated urine is being made. So when there is a dehydration situation, active aquaporins in the collecting duct Here you can see, when there is hydrated, no aquaporins in the collecting duct. So when dehydration is happening, aquaporins is inserted in the collecting duct for the removal or reabsorption of the water, making the urine concentrated. However, in the hydration condition, no aquaporins are there in the collecting duct. Yeah. So vasopressin binds to the membrane receptor. Okay. So this is the collecting duct. Here you can see the vasopressin, which is coming from the circulation, is binding to the membrane receptor, which is making the cyclic AMP and making the secondary messenger, and from there the aquaporins vesicles are being integrated into the vesicle membrane or this luminal membrane. Okay, so these vesicles are integrated in the membrane. So the receptor activates the cyclic AMP, the secondary messenger system, and cell inserts aquaporin water pores into the apical membrane, and water is absorbed by the osmosis into the blood. Also same thing. Yeah. So again, so recirculation of urea is also happening. With this, osmolarity is also being maintained with the help of urea. So recirculation of urea, so this region which is you are able to see here, that is impermeable for urea. Okay, so when the urea is coming into the filtrate, it is 4.5 of the total uh, uh, substances which is making the osmolarity. Then as it is going further in the renal tubule, only 50% of that is being remaining, and because of the water reabsorption, it is being concentrated. Okay, then in this portion it is impermeable for urea. Okay, then in the end of the collecting duct, it is permeable to urea, but it also crosstalk with this particular section in the intermedulla, and that also makes the osmolarity of the filtrate. So urea also plays an important role in the formation of concentrated urine. A protein-rich diet leads to increased urea production, thus increased urine concentrating capacity of the kidney. So during the periods of water deprivation, the kidney develops a steep urea gradient from the cortex to as much as of 100-fold higher in the inner medulla. So while the active transport of ions out of the renal tubules is currently thought to provide the main energetic drive force for creating this gradient, passive transport through urea transporters also contribute through a counter current exchange mechanism that shows the diffusion of urea away from the inner medulla. Yeah. So this is acute regulation of urea transport by vasopressin. So vasopressin also helping for the urea transport. So when the body is dehydrated, ADH is released and bind to the B2 vasopressin receptor located in the basolateral membrane and activates this particular protein, and this increases the intracellular cyclic AMP synthesis. So both are involved in the vasopressin and in urea permeability. And also the next mechanism is thirst. So drinking is regulated by plasma osmolarity and the extracellular fluid volume in much of the same fashion as vasopressin secretion. So water intake is increased by the increased effective osmotic pressure of the plasma, by decreased ECF volume, and psychologic and other factors. So here you can see vasopressin. So in plasma vasopressin as the and this is the plasma osmolarity. So as the plasma normally the osmolarity is 280, 260, 285. Okay, so as the plasma osmolarity is increasing, with the increasing the plasma vasopressin level is also increasing. Okay, and that is also furthermore increasing the thirst, which is the physiological sense. So vasopressin is a nanopeptide synthesizes a prohormone by the hypothalamic neurons and released at the posterior pituitary, and its half-life is about 16 to 24 minutes. So effect of plasma osmolarity in the plasma vasopressin. Okay. and this is the part which is the diabetes insipidus. So this refers to the high rates of production of dilute urine, so either because of the posterior pituitary fails to release the vasopressin or because the kidney does not respond to this particular hormone. Okay, so this should be the contrasted with diabetes mellitus, in which increased urine flow is consequence of the osmotic diuresis. Okay, because the vasopressin is not acting or maybe the receptors are not working, because of that the diuresis is happening by the filtration and the incomplete reabsorption of large amount of glucose. So the failure of produce or release vasopressin is referred as central diabetes insipidus, and this is a rare congenital disorder, but it may also be caused by the surgical procedures. Okay. Removal of the pituitary does not always cause a total lack of vasopressin production, but in some of the nerves responsible for releasing the hormone and in the pituitary stalk are not lost. Okay, so the treatment for central diabetes insipidus is administration of a synthetic analog of ADH, which is desmopressin, which acts selectively on V2 receptor to increase water permeability in the late distal and the collecting tubule, and the desmopressin can be given by injection as a nasal spray or orally, or rapidly restores urine output towards the normal. So it is a synthetic replacement for vasopressin, the hormone that reduces the urine production, and it may be taken nasally, intravenously, or an oral sublingual tablet. Okay. Then this is a hormone of atrial natriuretic peptide. So it's a powerful vasodilator, okay, and a protein hormone secreted by the heart muscles. Okay, so in the atria, okay, natri because it is related with the sodium, and the uretic related with the urine, it's a peptide hormone, so it is atrial natriuretic peptide. So it is involved in the homeostasis control of the body water, sodium, potassium, and fat. So it is released by the muscle cells in the upper chamber of the heart in response to high blood volumes. Okay, so when there is a very high blood volume, that blood is coming to the arteria, right arteria, and that arteria is sensing in the arteria this particular hormone is being released. So specific cells of the cardiac arteria, when descendant because of the plasma volume of expansion, secretes a peptide ANP and acts to reduce the water. So sodium and adipose loads on the circulatory system, thereby reducing the blood pressure. So ANP has exactly the opposite function of the aldosterone secretion by the zona glomerulosa. Okay, so that is aldosterone stimulates sodium retention and ANP generates sodium loss. So ANP, what it does, so aldosterone was making more and more sodium transporters for the reabsorption. However, ANP inhibits the ENaC, which is the sodium transporter in the distal convoluted tubule. So it inhibits the ENaC, and that's why sodium reabsorption was not happening, and because of that water is also being retained in the filtrate, and that is why the volume of the blood is regulated. So increased level of this peptide in turn inhibit the reabsorption of sodium and water by the renal tubules, especially in the collecting ducts. So the decreased sodium and water reabsorption increases urinary excretion, which helps to return blood volume back to the normal. So these are the hormones. So from hormone, this ANP hormone is being released, which is helping the GFR. Then there is aldosterone, angiotensins, ANP causing vasodilation, decrease in the blood pressure. So let's see. So here so this is the vasopressin, the osmoreceptor activation. Okay, So when the osmoreceptor activation is happening, plasma vasopressin is secreted. Vasopressin receptor activation, water reabsorption, which is giving a negative feedback. Okay, so the third one here you can see is the pituitary gland. Okay, the pituitary gland. Then we have the first one and the second one, which is, so this is kidney. Okay, kidney and pituitary gland. And then the first one, which is the hypothalamus, and this is the second one, which is the plasma osmolarity, which is being sensed by the osmoreceptor activation. Okay, then this is the ADH secretion. Okay, so what is happening? So the signal which starts from here, the fourth one. So a lot of water is being excreted from the body. So when the water is being excreted, H2 excreted, then the feedback goes that the water deficiency is there. So water deficient feedback goes. So when that feedback goes, what happens? The plasma ADH level increases. Okay, the plasma ADH level increases. So when the water osmolarity is there, more water deficiency is there, then the osmolarity increases. So the extracellular fluid osmolarity increases, and that is sensed by the osmoreceptors. And because of the osmoreceptor, ADH secretion from the posterior pituitary increases, which overall causes more ADH in the plasma. And because of that, water permeability in the distal tubule and collecting ducts is being increases. Okay, because ADH is implementing the aquaporins in the collecting ducts and the distal tubules for the permeability of the water. Then water is being reabsorbed, and then if there is a excess excretion of water, then again the signal of water deficiency goes to the brain. Okay. Yeah. So here we have the signal. So this is the set point, the normal osmolarity, where the vasopressin is being released. So normally our cells are having the minus 60 millivolt voltage. Okay, then when there is a hypertonic situation where there is a lot of osmolarity, then in that condition lot of sodium ions are there, and because of the high sodium ions, the membrane is depolarized. Okay, the membrane is depolarization is happening. So the because of the presence of positive ions, the membrane potential from minus 60 it goes to minus 55. Okay, so because of the depolarization, the action potential frequency increases. With lot of action potential frequency, vasopressin increases. So when the vasopressin increases, it causes the antidiuresis. Okay, and then the other condition, when there is a hypotonic condition, okay, when there is a less sodium means the millivolt or the plasma inside the nephron potential, which is minus 65 volt, it becomes hyperpolarized, more negative, because of the less sodium ion present. So it goes to minus 65 and produces and that shows less action potential and low vasopressin. So that causes diuresis to bring it to the set point. So diuresis, removal of more and more water, bringing it to the set point. Okay, so here it has been shown the osmolarity. This is the plasma osmolarity which is being shown. So as the plasma osmolarity increases, the vasopressin in the plasma increases. Okay, so because the plasma osmolarity, vasopressin is sensed because of the high plasma osmolarity, it is sensed by the osmoreceptors, and that osmoreceptor increases the vasopressin production and increase in the plasma vasopressin. Okay, and as a feedback to that will be thirst, because your body is in need of water because of the high osmolarity. So that gives a psychological signal, that is thirst. So as the osmolarity increases, plasma vasopressin level increases, and also the thirst increases, which again causes the consumption of water, which is here. Okay, more thirst, drinking of water again. When the water is you drink the water, then the signal goes to the brain, and then the signal goes to the the plasma osmolarity is restored. Okay, so that is how the entire process takes place. Okay, so vasopressin causes the water reabsorption. Okay, so that is how it, and when the water reabsorption is happening, it again gives the signal to the brain. So there are two mechanisms. So osmoreceptors gets activated when there is a change in the osmolarity. Okay, so that change in the osmolarity is sensed by the osmoreceptors. They gets activated and they increases, they follow two pathway. One is the they increase the thirst. Okay, and another one, plasma vasopressin level. In the plasma vasopressin, it causes the water reabsorption, and when the water is reabsorbed, it again sends back to the brain. And similarly, the thirst is another response, which when you drink the water, that also give the signal to the brain that now the osmolarity is restored, and that is how the cycle takes place. I hope it's clear. Okay, I hope it's clear. If you want to ask me anything more, you can ask. Okay,
No, ma'am.
okay, if there is no question, I'll conclude today's session. See you next week.
Thank you for your class. Thank you, ma'am.